Okay, so we're going to talk about the blood. So some of the functions of the blood include transporting various items such as oxygen, nutrients, sugars, lipids, waste, and hormones. Okay, so it also maintains and regulates lots of things. So it maintains and regulates the temperature, it regulates the pH of the body, it maintains and regulates fluids, and it also helps, there we go, and it also helps with protection. So it helps protect from infection and it protects from blood loss as well. Now, if you took blood and you put it into a test tube and you spun it in a centrifuge, it would separate into three main areas. So here is the test tube. All right, so this bottom layer here, there, is the red blood cells. So I'm going to color it red. There you go. You're welcome. That is the Buffy band, which includes white blood cells and platelets. Then the bigger band above is the plasma. That includes water, gases, and nutrients. Okay, so next we're going to talk specifically about the Buffy bands. All right, so if you remember, the Buffy bands are composed of the the thrombocytes and the leukocytes. The thrombocytes are platelets, which the function is to help with clotting. And the leukocytes are the white blood cells, as you remember, but they have a lot of different sections. So the main area is they're broken down into granulocytes versus agranulocytes. The way to remember it is granulocytes have these big nuclei depending on what type it is, but they're speckled. Those are the granules. So they're speckled like in that picture. So they have the last name of Fils, and agranulocytes have the last name of Cyte. That's a way to remember it. So there's neutrophils, eosinophils, basophils, monocytes, and lymphocytes. Those are the 5 types of leukocytes. These 3 are granulocytes. Neutrophils, remember, are the most common. They are like the first responders. They're the ones that are first on the scene. They're multi-lobed nuclei, and they fight with bacteria. Eosinophils, they attack antibodies. So they typically fight parasites or allergies. And they are bilobed. And you'll see in the pictures in your lab packet what multi-lobe versus bilobes. Basophils produces inflammation. They're rare and they have the nuclei have 1 lobe. So the agranulocytes, there's a monocyte and a lymphocyte. The monocyte is a big mammer jammer. It engulfs the whole bacteria. And the nuclei is a multi-lobe. The lymphocyte has different sections too. It has T cells and B cells. Typically, with the helper T cells are low, it would be HIV related, and mono would be with atypical lymphocytes. So you can see that eosinophils are the only ones that are bilobed. You have 2 that are multi-lobed and 2 that are 1-lobed, but you only have 1 that's bilobed. So one thing for some reason didn't work when I tried to record was about anemia. So anemia is low red blood cells. There's a lot of different types of anemias, but I just wanted to touch on a few of them. For instance, iron deficient anemia. So if there's not enough iron right here, then there's not enough oxygen, there's not enough space the oxygen can bind to it. If the oxygen can't bind to the iron, then this hemoglobin's going to be desaturated. If the heme group will be desaturated. If the heme group is desaturated, then this hemoglobin will be desaturated. If the hemoglobin is desaturated, what is happening? Hang on.
Okay, this next section is on blood typing. ABO, blood typing. So I specifically didn't speed up what I'm drawing because this is very confusing at times and I want to make sure you fully understand what I'm saying. So in each cell, there is a cell membrane, right? We'll say this is the cell membrane. There are certain antigens that come off of the cell membrane. So we're going to say this antigen looks like this. It might not, whatever. So that is an antigen. Okay, that's an antigen. We're going to talk about antibodies and antigens. So this is an antigen. Okay, so the possible types, now this is only for blood typing. There's a zillion types of antigens out there, but the specific types for blood typing, there's different types. There's antigen A, antigen B, and antigen RH. Okay, so each cell in our body has whatever our blood type is, whatever that antigen is, whatever our blood type is has that antigen. So when we're talking about ABO, there are 4 different blood types. There's type A, there's type B, type AB, and then type O. Okay. Type A will have a cell that looks like this. We're going to say this antigen is A, and give me a different color. B, this antigen will be B, and RH will be orange, because RH sounds like orange. Whatever. Okay, so a cell that is type A would look like that with little red antigens all over it. Okay. Type B, you see where I'm going with this, would look like this. Right now you're thinking this is not hard. I know. Again, with B antigens. All right, so this has A antigens, this has B antigens. Just because I have to do that kind of stuff. Okay, type AB will actually have both. So it'll have type A antigens, and it will have B antigens. Type O have no antigens. So now you know what the cells would look like in a person with type A, a person that's type B, a person that's type AB, and a person that's type O. Here's the thing. If a person has ever come across any other antigen, what happens is your body makes antibodies to that antigen. So if an antigen comes in, we're just going to grab a different color. Here's purple. Okay. If an antigen happens to be on the cell, that is, I have no idea, Penn State antigen. Okay. This is a new cell. We don't know who this is. This is a new cell that has come into the body, but we don't know what that is. So we're going to make an antibody against it. This antibody will fight against that particular antigen. Now, if it's our cell, then we are used to it. We will not make antibodies for ourselves. We only make antibodies for foreign antigens. Okay? So here we have A antigen. So we're not going to make A antibodies. That doesn't make sense, right? Because that would just kill our own cells. So what we do instead, someone who's type A will make anti-B antigens, or antibodies, I'm sorry. Oh my goodness. So here they have B antibody and A antigen. All right. The next one has B antigens already on their cell. So the B is not foreign, so they're not going to make B antibodies. They're going to make A antibodies. So here's A antibodies, A antibodies, A antibody, B antigen. Now here we have the B antigen and the A antigen. So what kind of antibodies do you think they would have there? They're going to have no antibodies there. They're not going to have any antibodies against A because that'll kill their own cells. They're not going to have any antibodies against B because that'll kill their own cells. And those are the only two that we're talking about right now. Now for type O, they have no antigens, A or B. So they need a lot of antibodies. So they're going to make some for B because B is foreign, and they're going to make some for A because A is foreign. A antibody and B antibody. All right. So I'm just going to go through that again because we're going to get complicated because we're going to talk about transfusion and stuff like that. So again, type A will have A antigen and will have B antibodies. Type B will have B antigens and A antibodies. Type AB will have both, and type O will have no antigens, and therefore they have a ton of antibodies. So you know these antibodies will destroy anything with those antigens. So if I tried to give this little critter to this person, put this cell in here, what do you think would happen? These things would fruit ninja and slice this thing in half. Okay? So we can't give type B blood to someone who's type A because these antibodies will kill that cell. Does that make sense to everyone? Not that you can answer me, but if it doesn't make sense, you can always finish watching the video and then ask me. So, all right, so let's talk about transfusions. So what can we give type A? What can we give type B? What can we give type AB? And what can we give type O? So, I know my boxes get annoying because I draw them all the time, but I'm not really that sorry. Okay, so we're going to start first. Let me draw my little circles here. All right, we're going to start first with type A. Okay. type A. So can we give this cell to this person? Sure. Because these antibodies are not going to attack anything on that. So it's okay. Yes. Can we give this cell to this person? We actually cannot. These antibodies will attack that cell and kill it. We'll slice it right in half, cut it right in half. This is a no. Can we give this cell to this person? Sure. There's no antibodies to kill it. Can we give this cell to this person? These A antibodies will kill that. So no. If you didn't follow that, probably within the next 3 you will. So here's type B. I'm just writing these real quick. Okay, so type B with these little nubbins. Can we give this cell to this person? No way. These antibodies will slice that puppy right in half. No. Can we give this cell to this person? They have antibodies, but it doesn't affect that cell, so who cares? Yes, sirroo. Can we give this cell to this person? Sure, there's no antibodies to kill them. Can we give this cell to this person? This has the B antibodies, which will kill it, so no. All right. Now you have type AB, which is both. How fast can I color? Okay. All right, can we give this cell to this person? It has these antibodies against these antigens, it will kill it, so we have to say no. Can we give this cell to this person? These antibodies will kill this cell, so no. Can we give this cell to this person? Sure, there's no antibodies. And can we give this cell to this person? That would be a bad idea because those antibodies are going to attack it. No. So for AB, it can only receive. So it is the universal recipient. Okay? It's the universal recipient. So type O looks like this. Can we give this cell to here? I mean, it has those antibodies, but there's no antibodies on here, so sure. Can we give this one to here? Yes. Can we give this to here? Yes. Because there's no, it doesn't matter what antibodies they have. Type O has no antibodies on it, so it doesn't matter. And this is a yes. So type O is a universal donor. I don't want to put it there because that's kind of confusing. So when I say universal recipient, I mean you can accept blood from every type. So someone who's AB can accept blood that's A, can accept blood that's B, can accept blood that's AB, and can accept blood that's O. Someone that is a universal donor can give blood to everyone. So type O can give it to A, can give it to B, can give it to AB, and can give it to O. So the universal donor is type O, the universal recipient is type AB. However, there's one more antigen we haven't discussed, and that's RH. So there's a few different blood types. There's actually 8 blood types. Right now you're thinking, but I only saw 4. 1, 2, 3, 4. There's A positive, A negative, B positive, B negative, AB positive. Do you see a trend? AB negative, O positive, O negative. And my box, because I have to. That's what I do. And arrows. All right. So for the RH, You only have two options. It's there or it's not there. Okay, so if something is Rh positive, then that cell, whatever that cell is up here, will also have some orange on it. Okay, so if it's A, it would have red and orange. If it's B, it would have blue and orange. If it's AB positive, it would have both of those in orange. If it's O, it's just orange. Now, because this is Rh positive. Rh positive. A cell that is Rh negative is going to look like that, other than whatever blood type they have. However, if you're Rh negative, you're going to have a ton of antibodies against that antigen D. Oh, I didn't tell you that. The Rh antigen is actually called antigen D. They're used interchangeable. So, let me put it here. Antigen D. All right. Antibody D. So, can you give someone who is Rh negative an Rh positive cell? You know, of Rh positive blood, whether maybe it's A positive or whatever. You can't, because it would lyse the cell in half. So what happens is these antibodies attach to the antigen and cut it down and kill the cell, which is what it's supposed to do, but not for, you know, changing the blood type. Okay, so there's that. All right, so now you know all the different types of blood types. But you guys are going to actually determine someone's blood type. Determine blood type. Well, I mean, I guess technically my kids determined it, but you guys are going to evaluate the results. All right, so you're going to watch a video of my kids mixing some blood and some droppers and then adding some antibodies into it, and then you'll see the results. If antigen mixes with antibody, agglutination occurs. Okay, so agglutination is when it kind of clumps. All right, so you'll have lysis, you'll have clumping. It's trying to kill that cell. So if this antigen, we'll say B. Why not? Mixes with this antibody, then you're going to get clumping. What that means, in the vial, when you look at the blood type, you're either going to see something nice and smooth or you're going to see something speckled. This would be something speckled. If there's no antigen, then there will be no response if you're adding that antibody. So what we're going to do is we're going to take vials. So this would be A, this would be B, and this would be D, which is that Rh. So we're going to take a drop of blood, drop it in, drop it in, drop it in. And then we're going to take, I need some colors here. Let's say we're going to take antibody A, and we're going to drop it in here. We're going to do antibody B, antibody B, and antibody D, and drop it in here. Then we're going, well, you're not, but you're going to watch them mix each one. and you're going to see what happens. One of two things will happen. You're either going to get a nice, smooth response, which will look like this, or it's going to look more like this. So when you see this, you know that agglutination occurred, which means you know that there's that antigen in there. Because if there was no antigen, there would be no response. The antigen has to mix with the antibody for there to be agglutination. Okay, so if this had no antigen A, if it mixed with antibody A, it wouldn't make a difference. So it would be nice and smooth, right? So if this one was, we'll just make this one spotted. Okay, just so you know, A, B, and D. So this one is nice and smooth. So therefore, we know there's no A antigen. However, these are clumped, so we know there is a B and D antigen. Therefore, you know the blood type is B, because B is positive, or B is, I shouldn't say positive because that's what I'm about to say. And then because the antigen D is there, it is Rh positive. So the blood type for this one would be B positive. All right, I hope that made sense to you, because this is a hard concept to understand. When you see this, that means that there's an antigen. If there's an antigen, it's going to be in the name. That should help. All right, real quick, a genetics review. Because one of the experiments that we do is figuring out whose baby belongs to which mama. Yes, I said that correctly. All right. So some of you already had genetics. If you did, ignore it, and you know this already. This is way basics. However, if you have not had genetics yet, this is just so that way you have a basic knowledge about it. So there's phenotypes and genotypes. The phenotypes is what we see. The genotype is the genetic info. So it's 2 alleles. Alleles is that particular gene. So you get one from mom, one from dad. So, boo. All right. So for phenotype, we know that they can be type A, type B, type AB, or type O. That's it. Those are the phenotypes for blood types. Type A, I mean, technically it would be positive or negative, but we're going to not go there with that. Type AB, type B, and type O. Now we get one from mom and one from dad. So for type AB, you know that the allele, one came from A, or mom or dad gave A and mom or dad gave B. So the only genotype it could be would be 1A and 1B. So mom or dad gave that one, mom or dad gave that one. O is a lack of 1A or B. So we use a little I for that, or little, I don't know how you want to say it, but. So O would be this, because it doesn't have an A and it doesn't have a B. Now, A is dominant. So this could be either be this, 1A, so we got that from mama and that from daddy, or that and that. So if you ever see this, it's almost like the person doesn't even have this. It doesn't exist because there's no difference between these two. But you just don't know. You don't know which one, if someone's type A, which one they are. So IB, B, or big I, little I. So type O is the lack of A or B gene. Type A could either be dominant A, big A, big A, big A, little I. AB is only big A and big B. Type B is big B, or big B, little I. All right? The reason why I want you to know this stuff is because you need to put it into a Punnett square. Ever heard of that before? Some of you guys are probably shaking your heads like, yeah, I do. Some of you guys are completely clueless because you haven't had it yet. You will. All right, so Punnett square looks like this. Neat. So then you put mom's alleles up here. And you put dad's down here. Okay? So let's say that the patient or the person in question is, I don't know, let's pick this one and this one. Okay? So mom is type A, but you know, for some reason, you know that it's that one, just to show you. So you put big A, little I, and then we're going to do this one. All right, so daddy gave that one. Dad's type AB. So then you cross them. 1A, 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 I, 1A, 1B, 1B, I. All right, so what happened is if mom and dad had 4 babies, theoretically, they would each be one of these blood types. So if they had 4 babies, this one is type A. This one is type A. This one is type AB. And this one is type B. So in theory, 25% would be type B, 25% would be type AB, 25% would be heterozygous, meaning the two are not different. 25% would be homozygous, which means the two are the same. So 50% chance of type A, 25% chance of type B, 25% chance of AB. That's how you would figure out mom. Okay, mom, who's the mom, who's the daddy, etc. So I want to do another one of those. Just practice a minute, because if you haven't ever had to do this before, it can be a little confusing. If you have done this before, you can kind of fast forward just until the very end. So, because I just want to give you a few more examples. All right, so let's say both mom and dad have AB blood. Okay, so mom has AB and dad has AB. So here's 1A. Whoa. All right. 1A, 1A, 1, oops, A, 1B, 1A, 1B, 1B, 1B. So, how many of these, how much chance do you have of having someone who's type A? You have 25% chance because it's 1 out of 4. Oops, I forgot to write type A. A type. B type would be 25% as well because there's only 1 out of 4. However, there's 2 out of 4 chances to have type AB. So that would be 50% because you have 2 out of 4. All right, so can mom, who is type A, and dad, who is type B have a type O baby? That's the kind of question that I might do. Okay? Not particular this one, but just this in general. All right? So let me bring this back. I might have to turn this light off. Whee! This is so neat. I'm using my daughter's toys. Okay. So here are the different options. So for type A, I wonder if you can see it. Yeah, good. So for type A, you know that it's either this or this. For type B, you know it can be either this or this. But in order for the baby to be type O, you need two little eyes. So you know it can't be here and it can't be this. Because if you had, you know what, let me just show you. Hold on. Let's say you thought maybe it could be this. We'll do that one and this one. All right. So here, 1A, 1A, and we'll do little I. So the combination for this, none of this has that, correct? You can answer me. I hope you're shaking your head yes. I think I hear the rattling. Okay, so since we know we need a little I from, essentially a little I from mom and a little I from dad, let's take this one and this one. So 1A, little I, 1B, little I, 1A, 1B. Now we combine them. 1B, little I, 1A, little I, and then little I's. This way, yes, the mom and dad with a blood type of A and a blood type of B can have a baby who is type O. So that is that. If you are confused by this, this is not an easy concept. I swear to you, it is not easy. So if it is confusing to you at all, you have two options. You could email me and we can set up a Zoom privately, or we can meet on Tuesday from 12 to 3, either one. Make sure, I don't know if I'm going to add it onto here, so there could be a video. This could be just in the middle of the video, but there's a video of blood type and of the blood type experiment. And also there's going to be a video explaining about something called hemolytic anemia of the newborn. That basically is trying to figure out if mom is Rh negative and baby's Rh positive. And remember how I said that if these two combine, then it can kill the cell, like fruit ninja slice through the cell. So there's going to be another video of that too. If it's not attached to here, it will be, I don't know, a link will be sent to it. So there you go. So I will see you guys on Tuesday if you want. If not, email me. On a side note, you guys did awesome for your lab packet, the first one. Everyone that turned it in did amazing. I think everyone got a B or above, so excellent. Good job. And that's it. Okay.
Okay, I can hear the chewing, weirdo. All right, so we're going to talk about hemolytic anemia of the newborn. That is when mom is Rh negative, but baby is Rh positive. So here is my blood cell, okay? I'm hoping that it is correct with you guys looking at it, because it's backwards for me, but it says type A negative. Now, with my first baby, this one who doesn't want to be on camera right here, even though I really, really want her to be, but that's her beautiful hand. Okay, so this one, so the first baby is A positive. Then what happens is at some point during delivery, the blood will mix. And so we will the blood cell will come into our world. Now, if you notice, if I'm A negative, I don't have these big Rh antigens, correct? So I end up making giant antibodies against those antigens. It doesn't affect baby number 1 because she's already born and out of the womb and going off and going into 6th grade. So it doesn't really affect her. But then the next baby comes along. Next baby, and you can actually stand up. There you go. She is also A positive, but now here I am with my little A negative blood cell, but now I have antibodies. So that means the antibodies then, when these antibodies can go through the placenta and go into where the baby's blood is. Yes. Okay, hold on. Hold this out. Finds that, combines it, and then like we talked about in the other video, Fruit Ninja slices that thing in half, which is really bad because it's called hemo, red blood cells, lytic slicing of anemia, low red blood cells of the newborn. You are the newborn. Yes, even though you're not that new. So that's why this is called hemolytic anemia of the newborn. So it doesn't affect baby 1, but it does affect baby 2. There you go. I hope you understood that. Say goodbye.
Bye.